What's up, guys? We're back, another educational video, and this week is a new study, the death of protein distribution. But first, make sure you like the video, subscribe to the channel, and leave a comment for the algorithm. New study that got a ton of play in the fitness community came out of Luc Van Loon's lab with Jorn Tromelin as the lead researcher. Van Loon is a very, very well-known protein metabolism researcher. He's been around for decades. One of the peers of my PhD advisor, Dr. Don Layman. Basically, people are saying this study shows that protein distribution doesn't make a difference on muscle mass. I have actually kind of beat the drum about protein distribution because we know, and there was a paper published by Alan Aragon on this, that around 25 to 30 grams of protein will maximize the muscle protein synthesis response after a meal. And if you just pump in more protein, it doesn't make the response go higher and it doesn't appear to make it go longer. People like me have said, if that's the case, if you are stimulating protein synthesis and you're better off getting multiple stimulations of protein synthesis per day compared to just one, so it probably makes sense if you're eating, say, 200 grams of protein a day, that you're splitting that up into a few different doses. Because if you're eating 100 grams of protein, you're probably not getting more protein synthesis than 50 grams of protein at that meal. So this new study was looking at the protein synthetic response to either 0, 25, or 100 grams of milk protein after a resistance exercise bout. What was really cool about this study, and this study was very involved, what was really cool about this study was they used what's called an intrinsically labeled milk protein. So in order to measure muscle protein synthesis, I'm going to give you guys the abridged version, but what you do is you take a stable isotope labeled amino acid, like phenylalanine, which is what we did. So this is not normal phenylalanine. It is labeled with a stable isotope that you can separate out on a GC mass spec. And what they're looking at is, okay, that label, we can see how much is in the blood, we can see how much is in the intracellular fluid, we can also see how much winds up in the muscle tissue. And we can use those different pools and how much is in the tissue to estimate the rate of muscle protein synthesis, because you can assume that all of the label that winds up in the muscle got there because of protein synthesis. They used a multi-label study, so they had several different amino acids that were labeled, and one of them was actually intrinsically labeled in the cow's milk. So usually the way you get a label in animal studies is you infuse it or inject it in the tail vein. In humans, you infuse or inject into the bloodstream to get that label in there. But you could also see more cool stuff like rate of appearance and whatnot into the bloodstream if you use an intrinsic label. Now, what's an intrinsic label? It means they took cows, infused them with this stable isotope, and then when the cows produced milk, that milk had the stable isotope in it. Super cool. Very involved. They had these subjects show up, do a blood draw, do a biopsy, do a resistance training session, do another blood draw, biopsy, then had them do blood draws and biopsies at other time points. So they measured a bunch of different stuff over a 12-hour period after eating either 0, 25, or 100 grams of protein. And again, very involved study because after these people finished resistance training and drank their protein shake, they had to lay down and basically just like lay there for like 12 hours. So this is very involved. Again, when people are like, why wasn't there more subjects in the study? Because good luck getting people to lay down for 12 hours after you've stuck a bunch of stuff in them and resistance trained them very hard, and then you're taking chunks of flesh out of them and blood samples. Like, good luck. Okay, good luck. You go do it. And so they found that between 25 and 100 grams of protein, 100 grams of protein produced a significantly greater overall effect of muscle protein synthesis. They showed a bunch of other stuff too. But basically they showed that, hey, there's supposed to be this cap at 25 grams. We didn't see that in our study. The 100 grams was way better. And the duration of protein synthesis seemed to be extended with the 100 grams compared to 25, which is in opposition to the studies I referenced where they show kind of a defined period of time that protein synthesis runs, and then you have what's called the muscle full or refractory effect. So a lot of people have said, well, see, look, like you could just eat all your protein in one meal if you want, or have just a big dose of protein. There's not like this upper limit on things. And, there is some teleological evidence for this. There are some things that I've had a hard time reconciling, like, okay, why would the body evolve to be like this? Because if you came across like a woolly mammoth or something like that, just caveman days, you can't keep the protein around. You've got to eat what you can eat, and then you're done. So from that perspective, it makes sense that there wouldn't be a cap on this stuff. But then when you look at the research data And even my study in grad school, where I did show that protein distribution had a small effect on muscle mass in rats, but again, rats a good model for protein metabolism in humans, I thought, okay, well, this shows, pretty decent evidence that perhaps protein distribution is important. Full disclosure, I talked to my PhD advisor, Don Lehman, about this paper. I also talked to the lead author, Jorn Tromelin, about this paper. We all agree that the big difference in this paper is these were untrained or not really trained individuals, and they did a pretty challenging resistance training session. I think what this shows is when you do a challenging resistance training session, you increase the demand for protein. Resistance training on its own will increase the muscle protein synthetic response. And in untrained people, we know that it can be up to over a few days in response time even. Certainly more than 16 hours, I know that for sure. They're measuring this 12-hour window. And so if you're increasing the requirement for the substrate of protein, perhaps there isn't that cap like we previously thought. But if you're somebody who hasn't resistance trained, or you're maybe very well trained, and we know that people who have been resistance training for a long period of time, their duration of muscle protein synthesis is much, much shorter compared to people who haven't trained or are untrained or minimally trained. I think perhaps as you get more trained, maybe distribution matters a little bit more, because you aren't creating that huge demand at a given time for that protein source. And I think the other thing here is the 25 grams, I'm not sure I necessarily agree with that. I don't want to go back and Monday morning quarterback the researchers, because this was actually a really great study, I think. But 25 grams of protein from milk protein is 80% casein. And that is a relatively slow protein. And there are some studies showing that to maximize anabolism with casein at a given time, it may take a greater amount compared to other proteins because it is such a slow protein and it does not cause a rapid increase in plasma levels of leucine and essential amino acids. So I'm curious as if they'd use 50 grams of protein, which we do know would absolutely max out the response, would that be different than 100? I'm not sure. Maybe it would, maybe it wouldn't. But I do think it's a question worth asking. But again, they have limited subjects. They picked what they picked, and it's very easy to go in and Monday morning quarterback a study and say why they should have done it differently. But I do think the resistance training is the key difference. And I think, again, if you put a gun to my head and inject me with truth serum and make me tell you what I think, my opinion is that I think this study starts to tie together some of the things that I'd observed, especially in like Grant Tinsley's research on intermittent fasting, where he had people intermittent fast and resistance train and still showed they gained the same amount of lean mass as people who were continuously meal feeding. But he did have them resistance train in their feeding window. Okay, so in that 8-hour feeding window, and he had them have at least 3 doses of protein during that time. So I think this kind of ties together that. I think what it says is total protein intake during the day is by far the most important determinant for dietary muscle protein anabolism. But I would stop short of saying that it makes distribution irrelevant. I still think that, especially for trained people or people who aren't resistance training, distribution may make a small but significant impact. I could be wrong on that. I think this is a great paper. I think it starts to tie together some of these things we see. I would love to see this experiment repeated with a couple different doses and in people who aren't resistance training, just to see if there's differences. But I think the takeaway from this is you don't have to be so concerned about distributing your protein perfectly evenly. If you have a meal of a higher amount of protein, it's not like your body is going to just waste it or waste all of it. But I still do think if your goal is just to build some muscle, then just make sure you get enough protein in during the day. If your goal is to be like me, which is to be the strongest, most muscular human being you possibly can, then I would still try to focus on getting multiple protein feedings per day. I think that will still be a little bit better than like one massive protein feeding per day. If you like these research breakdowns, make sure you sign up to my research review reps. Every month we break down 5 studies in fitness and nutrition. We break them down in a way that's easy to understand for anybody. And when you sign up, you get access to all of our previous issues as well as our how to read research guide. It's a phenomenal product. It's only $12.99 a month. And we help you separate out the BS from the legit stuff. So make sure you click the link in the description and go check that stuff out. I'll catch you guys next week.